concepts in molecular biology. Now we have already discussed these are the components involved in molecular biology: DNA, RNA and proteins. And these components, they together follow different processes which contributes to the central dogma of molecular biology. Gene, what is gene? Gene is unit of heredity which transfers genetic information from parents to their offsprings. The DNA segments that carries genetic information are called genes. It is normally a stretch of DNA that codes for either RNA or protein that has a function assigned in an organism. Genes hold the information to build and maintain organism cells and pass genetic traits to their offsprings. Now, so this is the chromosome. Now, as we have discussed, this chromosome is made up of DNA and proteins which help in folding of the genetic material into the nucleus. So this DNA, these are the segments shown onto the DNA which are known as labeled as gene A and gene B. The stretch of the DNA which codes either for RNA or protein. The gene can be defined as the region of the DNA that controls discrete hereditary characteristics, usually corresponding to single protein or RNA. This includes entire functional unit encompassing exons, that is coding region of the DNA, and non-coding sequences known as introns or regulatory sequences. Exons and introns represent coding and non-coding regions are present in eukaryotic gene. Introns are absent in prokaryotes. See, intron, the term intron and the term spacer elements are different, that we'll discuss later on. The introns are removed by splicing mechanism and the exons are translated in tandem to yield the functional polypeptide that undergoes further modifications known as post-translational modification to become a functional one. These functional polypeptides are targeted to various organelles in the cell and exported to the compartment where they are required. Either say when it is required to mitochondria it is transported, if it is required in chloroplast it is transported over there, if it is required in plasma membrane for functioning as transport protein, that particular protein is transported to that particular part where it is needed or where it functions. Now genome, what is genome? Genome is the entire set of genes, that is entirety of organisms hereditary information. It is encoded either in DNA or for many types of the viruses in the form of RNA. The genome includes both the genes and non-coding sequences. Genome includes both non-coding and coding regions, together known as genome. Genome is a complete set of genetic information of a cell, in particular complete sequence of DNA and RNA that carries this information. In diploid organism, it refers to the haploid set of chromosomes present in a cell. Depending on its localization, genome may be nuclear or organellar. Mitochondria and chloroplast, these are the two organelles which have their own genome. So genome is either nuclear or organellar depending on its localization. Organellar genome are of two types, mitochondrial and chloroplast genomes. Prokaryotes usually have only one set of chromosomes which is circular. Eukaryotes In eukaryotes, chromosomes appear in pairs. 23 pairs are present in humans. The cell containing them are called diploid. Since they have two sets of chromosomes, they are known as diploid organisms. The two sets of a pair are said homologous chromosome. Homologous since their height matches, their characteristics matches with one another, and each homologous pair, which consists two chromosomes, one chromosome is from mother side and one is from father. The genes which appear differently in the two chromosomes are called alleles. Alleles, these are different forms of genes present on these two homologous chromosomes, which one is from mother, one is from father. Cells which carry only one member of each pair are haploid. These are used in sexual reproduction. Haploid set is selected from each of the parent. Haploid set of chromosome is selected during the process of meiosis. Meiosis is reductional cell division. Not all the genes are expressed by specific cells. Okay. Not all the genes are expressed by specific cell. Few genes are silenced. They are not expressed by all the cells. The genome size is different in different organisms and different species of the same. The size of the genome governs the size and complexity of organism. However, many small sized organisms have bigger genomes than their larger counterparts. The genome contains all the genes present in the nucleus of the cells. All the genes are present in nucleus. Genes vary in size from few hundred to few thousand base pairs. Haploid set contain total genome of organism. Bacterium Mycoplasma genitalium has small genome size. Smallest genome is of Mycoplasma. 58 MB, and plant Triticum aestivum, that is rice. Rice has large genome size, 16,000 MB mega base pairs. That is 16,000 into 10 raised to 9 base pair. The genome size in human is 3200 megabases. Now see, this is the chromosome which contain genome of an organism, and see only this counterpart representing this segment. Okay, so this is gene say 1 and this is gene 2, and these two genes, they are separated by intergenic region or spacer DNA. Okay, the segment of the DNA which separate two genes from one another is intergenic region or spacer element, spacer DNA. These are comparative genome sizes of an organism. So you can see here, this we have already discussed. In human, size of the genome is 3.2 billion, but the gene number is only 25. Okay. Mouse, mus musculus, number of genes are same as in human and size is 2.6 billion. Okay. If you consider this plant, Arabidopsis thaliana, the size of the genome is 100 million, gene number is 25, chromosome number is only 10. Okay. E. coli, 4.6 million base pair, 1000 times smaller than the human genome. Size, gene number is only 3200 genes are present. Chromosome number is only 1. Since it is a prokaryotic organism, prokaryotes have only one chromosome, only one set of genome. They are haploid. Prokaryotes are haploid. Why to analyze genome? The prediction of the genes in uncharacterized genomic sequences. You can derive sequence and assign function to the genes to obtain complete sequence of many genomes as possible. You can sequence many genomes and keep those sequences in your databases for genome-modified plants, clones of species, likewise. Now the roles of genes within the cell. Gene contain instruction for each cell to make protein and RNA. Genes are made up of DNA fragments. Within the cell, DNA performs two tasks. The one, it acts as information repository, including instruction in making the component molecules of the cells, and it passes this information to the next generation. Okay. For example, say it is like a bank. It stores your money and it transfers it to whatever person you want to transfer. The mere presence of the DNA does not implicate the cell to be alive and functional. Mammalian RBC discard their nucleus. Genes are transcribed to the RNA. mRNA is translated to proteins. tRNA and rRNA serves as components of translation machinery. New functions of RNA are also being discovered, like regulatory RNA, miRNA, siRNA, and catalytic RNA. Proteins are structural components of cells, works like enzymes, hormones, various signaling molecules, receptors, and many more functions proteins carry out. For example, convert sugar to release energy and metabolite into the cell. Gene is the segment of DNA that codes for a functional unit. Housekeeping genes. A gene involved in basic function required for sustenance of the cell are known as housekeeping genes. Okay, these are expressed in all the cells. They carry out basic function, say for example cell division. The proteins involved in cell division, they are encoded by particular DNA segments or genes. These genes are known as housekeeping genes since these are required throughout the life of the cell. For all the cells, they are very important. Like we have to eat every day, drink water every day. Same way these housekeeping genes work. Housekeeping genes are constitutively expressed. They are always being expressed. They are never repressed or silenced. Luxury genes. Now the name itself indicates luxury. These are those coding for specialized functions synthesized usually in large amounts in particular cell type only. For example, now luxury genes are not expressed by all the cells. They are expressed only in specific cells or say tissues. For example, pancreatic cells, islets of Langerhans. Only these cells they produce insulin. No other cell in a body can produce or can secrete insulin. Only these cells. So this insulin gene is luxury gene which is expressed only in islets of Langerhans. Chromosomes. During cell division, the material of the nucleus became organized into visible threads which were named chromosomes, meaning colored bodies. Chroma means color. Okay. Material of the nucleus become organized into visible threads which were named chromosomes, meaning colored bodies. Introns. Introns, these are non-coding region of the DNA. Exons, coding region of the DNA. Junk DNA is this intergenic region between coding parts. Prokaryotes have little of it and eukaryotes quite more than 90% for the human genomes. Pseudogenes, genes whose sequences are homologous to those of functional genes. Homologous means sequences are more or less similar to the functional genes but which have accumulated severe mutations that render them non-functional. That is, pseudogenes are non-functional genes present in a cell. They do not have any function but they are maintained during evolution. Reading frames. One of the three possible ways of grouping bases from RNA sequences. Example, say this is the sequence TAA, TCG, AA, TG, GC. Now how can you read this sequence? You can read this in this way. First way is TAA, TCG, AAA, likewise. Second is you can start from the second nucleotide AATCGA, likewise. And third is you can start from this third nucleotide ATCGAA, TGG. Now you can see here these all reading frames are different if you skip say any nucleotide and start with the next one, and each one encodes different amino acid because each triplet encodes specific amino acid and each will lead to formation of different protein. Okay. Open reading frame. In a DNA sequence, a sequence beginning at start of codon having integral number of codons none of which being a stop codon. A human genome is blueprint of life. Contain coding as well as non-coding sequences which control all functions of the body. There are 20,000 to 25,000 genes present in human, which represent only 2% of the genomic sequence, while 98% is non-coding region. Remember, human genome is very much larger than other organisms' genome, but only 2% codes for protein and RNA. Rest of the 98% is non-coding. Non-coding regions include maintenance elements such as centromere, telomere, origins of replication which control DNA replication and repair, promoters, enhancers, silencers, and so on. So non-coding regions function in controlling cell, controlling element. It encodes controlling elements. Expression of genes is tightly controlled. It is crucial for genes to be expressed in correct cell type to appropriate level and at correct time. See, all the genes, whenever the signal is given, that particular gene is activated. Say in this pandemic, if we caught up with infection of COVID-19, our immune cells, they'll get a signal that yes, now we are corona positive. So our immune cells, they are activated and they try to produce antibodies against COVID-19 virus. Okay. So whenever required, only at that correct time, appropriate proteins are secreted. During cell differentiation and development, in response to internal and external signals, failure to regulate gene expression patterns can lead to serious consequences in genetic disease. Now this is central dogma we have already discussed. These we have already discussed, so we'll skip this. You can read this by pausing this video. Now see, wait. Now how the gene is transcribed. Now this is the genome. This is particular gene to be transcribed. Now this proteins, they bind to the sequence near the gene which initiate transcription. This sequence is known as promoter. That we'll discuss later on. From this gene, the RNA is synthesized. The process is known as transcription, and from the RNA, this RNA is processed. Then the proteins are synthesized. The process is known as translation. RNA synthesis, RNA processing, that is, if any introns are present, those are removed. At 5' and 3' side, the RNA is modified. It is splicing is removal of introns. Degradation of RNA is to control turnover of the RNA molecule. Unwanted RNAs are degraded. Then the proteins are synthesized from the mRNA template. Proteins are folded and processed. Post-translational modifications are carried out like glycosylation, hydroxylation, etc. And then the proteins are transported. Gene expression is either constitutive or inducible. Constitutive means housekeeping genes which are essential for sustaining life, thereby always continuously expressed. Inducible gene expression, whenever required, the signal is given for expressing particular gene and repressing particular gene. Now see, this is how the genes are controlled. The DNA, it is transcriptional control, that is, this process is inhibited, transcription process is inhibited. If the mRNA have to be repressed, the RNA processing control occurs. That is, either RNAs are not processed or degraded. If mRNA is processed, then RNA is degraded or the proteins are not synthesized. And if the proteins are synthesized, either degraded or activity of proteins are controlled. So the fundamental elements that regulate transcription. These are cis-acting elements, trans-acting elements, DNA protein interaction, protein interaction, and RNA polymerases. Cis-acting. Cis means to the same side. Cis-acting elements are the regions of the DNA or RNA that regulate the expression of the genes located on same molecule. Okay, often binding sites for one or more transacting factors, and may be located to the upstream side of the gene or to the 3' side of the gene, either in untranslated or untranscribed region. Now see, this 5' UTR, this is 3' UTR. This is exon 1, exon 2, exon 3. These are the coding regions. Interrupted in between by introns. These are non-coding regions. Introns are non-coding regions. These are removed. And then these exons are joined together for formation of functional RNAs. Okay. Now this promoter and this UTR, untranslated or untranscribed regions, they control expression of genes which are present to its side of the gene, same side of the gene. See, these are on the left side and this is on the right side of the gene. These are the examples of cis-acting elements. Promoter, DNA sequence where RNA polymerase binds and initiate transcription. Operator is the region. Regulatory sequence located immediately upstream of the structural gene that control transcription of an operon. Inducer located upstream of the promoter region, and DRS, downstream regulatory sequences. These are GC-rich sequences which have inverted repeats followed by 4-adenine signal termination of transcription. Transacting elements, these are located far away from the gene but can control gene expression. They have two domains. One is DNA binding domain and one is protein binding domain. Transcription initiation is tightly regulated process controlled by transacting elements both in pro and eukaryotes. Then the DNA protein interaction. There are few proteins which interact with the DNA and control or regulate expression of particular gene. So this one, see this green colored, this one and this one. These are regulatory sequences, and this is actual gene which have to be expressed, gene X. Now see, these are trans-acting elements since these are located far away from this gene. These are cis-acting. This TATA is promoter. This is located near to the gene, so it is cis-acting. And these are trans-acting elements located far away from the genes. Now to these trans-acting elements, certain protein binds which help in activating this particular gene by folding this DNA. See, this DNA is folded. Now see, अभी यहाँ पे कुछ gene protein bind हुआ है. See now this particular protein, which you can see this, like we play chess, that type of this protein it is. So this is one domain of the protein, this is another domain of the protein. This domain interact with the DNA and this interact with the another protein, see here, which is in contact with the protein controlling this gene. Okay, so this DNA is folded so as to interact directly with the gene to be expressed. To control the expression of this gene, the DNA is folded over, proteins bind and help in controlling expression of the gene. These are 4 types of the DNA binding proteins: zinc finger, helix loop helix, leucine zipper protein and homeodomain proteins. You need not to remember this. So these are the proteins which have 2 domains. One is activation domain and one is DNA binding domain. This activation domain binds with another protein, and this is DNA binding domain which binds to specific sequence. Then the RNA polymerase, key enzyme involved in transcription. Okay, in eukaryotes, RNA polymerase is of 3 types, which each one specifically synthesize single type of RNA.